if a recombinant DNA bearing gene for ampicillin resistance is transferred to E. coli cells and these host cells are spread on agar plate containing ampicillin. See, our DNA, recombinant DNA is having gene for ampicillin resistance. They are having ampicillin resistance. And if they are all culturing in a medium containing ampicillin, no problem. The host cells are now getting the ampicillin resistance. They are having an ability to grow in an antibiotic containing medium, ampicillin containing medium. So they will grow. So only a transformant. What type of transformant? A recombinant transformant will grow. Okay. Whereas non-transformant will not grow. So a transformant recipient cell will grow and non-transformant will die. See. This is the bacterium cell. And this is the vector. And this is the desired DNA. Now, this is the vector and this is the desired DNA. This has received the recombinant DNA from outside. Now it is called a recombinant transformant. Why it is a transformant? Because it has taken the DNA from outside without an agent. Transformation is a technique or a process whereby a DNA is entering into a host cell without an agent. We have studied bacterial transformation, Griffith effect, etc. Yes, same thing. So DNA has entered into, and that DNA is a recombinant DNA. It is having the DNA of our interest plus an agent, that is a carrier molecule, that is a vector. Now this is a recombinant transformant. This recombinant transformant is having the ampicillin resistance. This is having ampicillin resistance. Okay. So that it can grow in a cultural medium that contains ampicillin. So it will be growing. Whereas those who are not taken that recombinant DNA will not have the ability. Because E. coli, which is generally the host in our biotechnology of engineering, genetic engineering researchers, E. coli is having no natural ability to resist any antibiotics. By nature, this is having no ability to resist any antibiotics. Clear?